This video is going to describe the digestive system. Now, food is broken down in a process called digestion. The digestive system is made up of the alimentary canal, which is a muscular tube running through the body from the mouth to the anus, and also includes several other organs that need to make chemicals needed for digestion, and these include enzymes. First off, start at the mouth. This is obviously where food is taken into the body. Now, during chewing, teeth break up the food into smaller pieces. This increases the surface area for digestive enzymes to work on. The tongue helps to form the chewed food into a ball called a bolus. The bolus gets coated in saliva, which lubricates it and makes it easier to swallow. Saliva also contains an enzyme that starts to break down the starch in food. One of the enzymes in saliva is called amylase. Now, this breaks down starch into maltose. Next, the food travels down the esophagus. This is the muscular tube between the mouth and the stomach. Now, muscles contract in waves to squeeze the food down towards the stomach. This process is called peristalsis. Now, this diagram helps to show this process. Behind the food bolus, so the bit of food, the muscles contract. It pushes the food down. And in front of the bolus of food, the muscles are relaxed. And these muscles contract and relax in a wave-like movement, which is called peristalsis. The food will then reach the stomach. This is a muscular bag that makes hydrochloric acid and also some enzymes. One of these enzymes is called pepsin, which breaks down proteins. In the stomach, the food is churned up with the enzymes and the acid to make a thick paste of food. Next, the food will travel to the small intestine. This is a long coiled muscular tube where most of the large insoluble food molecules are broken down into smaller soluble molecules. It contains a lot of digestive enzymes made by the pancreas, and it also makes its own digestive enzymes. We'll take a look at these enzymes later on in these videos. Now, the molecules of digested food are absorbed into the blood using finger-shaped projections called villi. So if we take a look at this diagram here, the villi are these structures here. Now, the inside of the small intestine is lined with millions of these villi. On top of the villi, we also have microvilli. These villi and microvilli increase the surface area for absorption of digested food products. Notice the villi has a blood supply to each villus, so the digested products can go straight from the small intestine into the blood. Any undigestible food will then travel to the large intestine. Water actually diffuses back into the blood, leaving the waste material behind, which is known as feces. It will then travel to the anus, where the undigested food is passed out of the body. Another important organ in digestion is the liver. So, digested food is absorbed by the small intestine into the blood, and the blood will then be transported to the liver. Now, some of the digested food products are broken down even more, and some are built into larger molecules. The liver also makes bile, which helps in the digestion of fats. Now, bile emulsifies fats, giving it a larger surface area for the action of lipase, which breaks down fat. The gallbladder is a small organ that actually stores the bile. So the bile is made in the liver and stored in the gallbladder, and releases the bile into the small intestine when it's needed. Now let's have a look at some of the enzymes that are involved in digestion. The digestive system breaks down large insoluble food molecules into smaller soluble molecules. Now this chemical breakdown of food depends on digestive enzymes. Different types of digestive enzyme break down the three main types of food molecules: carbohydrates, proteins and fats. So let's start with carbohydrates. Foods like bread and potatoes are full of carbohydrates. Digestive enzymes that break down carbohydrates are known as carbohydrases. An example of a carbohydrate is amylase. Amylase is an enzyme that breaks down starch, which is quite a big molecule, into smaller sugar molecules. Amylase actually breaks down starch into maltose, and another carbohydrate called maltase breaks down maltose into glucose. So after starch has been digested into glucose, the glucose can then be absorbed into the blood at the small intestine. Next, proteins are digested by proteases. Proteins are broken down into shorter chains and then into amino acids. Pepsin is an example of a protease, which is made in the stomach. Now, pepsin works best in acidic conditions. The stomach walls produce an acid which makes it around pH 2 to 3. Now, this is actually the optimal pH for pepsin to break down protein as fast as possible. However, the contents of the small intestine are alkaline to neutralize the stomach acid. So the proteases released into the small intestine work best at around pH 8. This is shown in this diagram here, where the optimum pH for pepsin is around 2 to 3, whereas the optimal pH for another protease called trypsin, which is released in the small intestine, is around pH 7 to 8, so maybe just on the alkaline side. And now let's have a look at how fats are digested. Lipases are enzymes that digest fats. Now these lipases chemically break down the fat molecules into fatty acids and glycerol. Now fat and water don't really mix, so the fats and oils you eat form globules in the watery digestive juices. Now these large globules have a very small surface area to volume ratio. So for example here. This means the lipases can only break down the fat molecules very slowly. However, bile, remember, is made in the liver, stored in the gallbladder, breaks down these large globules into smaller droplets, here, forming an emulsion. We say that bile emulsifies the fat. These smaller droplets have a much larger surface area, which makes it possible for lipase to break down the fat molecules far more rapidly. More bile is released after a fatty meal. The bile from the gallbladder is actually alkaline. Now this helps to neutralize the stomach acid and produces the slightly alkaline environment for the protease enzymes of the small intestine to work in. So, for the exam, you will need to know the structure of the digestive system. You'll also need to know how digested food molecules get into blood. In particular, the role of the villi and the adaptations the villi have for absorbing digested food products into the blood. You will also need to know about the different types of enzymes involved in digestion and also the role of bile.